Thank you, Chairperson and organizers, for inviting me to this great conference. I will be talking some very basic things which may not have the interest of clinicians, but I will try to make the clinical correlation. The neuroendocrine regulation of glucose homeostasis. We know that glucose homeostasis is basically regulated by the endocrine glands, but the total homeostasis cannot be achieved by particularly one hormone or two hormones. It is always a combined effort, and we know for the controlling things there are only two basic control mechanisms. One of them is neural, another one is the endocrine. So both the neuroendocrine regulations are required for any physiological mechanism. We know homeostasis is a condition in which the internal environment of the body remains relatively constant despite changes in the external environment, and the examples can be the maintenance of body temperature or the pH or even the blood glucose. And when we talk about this glucose homeostasis, why it is important? Because glucose acts both as a source of energy and also a source of starting material for nearly all types of biosynthetic reactions. All biosynthetic reactions require glucose, and that is why the normal plasma glucose concentration had to be restored between 70 to 120 milligram per deciliter. This glucose homeostasis becomes more important because of the fact that brain has the high consumption of glucose, uses around 20% of the resting metabolic rate, and it is the first fuel for energy for the brain. And during exercise, the special thing occurs that the working muscle competes with brain for glucose, because at this time the muscle do not require any insulin. They can go to the glucose reaches the muscle without insulin. So at that way they are competing with the brain. Many redundant systems are available for maintaining the glucose homeostasis. They can be the hepatic glucose production and the pancreatic hormones, insulin, glucagon, amylin, etc., and the sympathoadrenal stimulations in the form of norepinephrine. And the glucose homeostasis, as we know, is more important here, and we know that the high blood sugar levels produce the insulin release, and that insulin goes to the tissue cells and make the glucose available for the tissue cells, stimulate glucose uptake from the blood, and stimulates the glycogen formation in the liver, and the blood sugar becomes normal. And by the same time, when the blood sugar level goes down, the glucagon is secreted from alpha cells, and that glucagon stimulates the glycogen breakdown and neoglucogenesis, and that again raises the blood sugar level. And that keeps on happening all the time. That is why that good homeostasis is required, and most of the time it keeps on happening. And any defect in any process will create the different type of diabetes. We know that it can be diabetes mellitus, or it can be adrenal diabetes, or it can be even pituitary diabetes. The endocrine regulation, we know this basically because of the insulin secreted from the pancreatic islet B cells, and release is regulated by the blood glucose level, and the blood glucose level is maintained in the threshold level. It stimulates the glucose oxidation and storage and inhibits glucose production. It also stimulates glycogen synthase. By that the glycogen is stored in the liver. It inhibits the phosphorylase so that the glycogenolysis do not occur. Inhibits the gluconeogenesis so that the hepatic production of glucose does not occur in presence of insulin. It stimulates glucose transport into adipocyte, which is then converted into triglyceride. It inhibits hormone-sensitive lipase and lipoprotein lipase. It activates the GLUT1. And release of insulin is inhibited by epinephrine and norepinephrine. Obesity increases, and training, exercise decreases the insulin secretion. And that we know from very basic things. And glucagon secreted from pancreatic islet alpha cells, it promotes liver mobilization of fuels, and that will increase the glycogenolysis and the new production of glucose in the liver. It stimulates the CMP, release is regulated by blood glucose level again, and activates the phosphorylase so that the glycogenolysis can occur, and stimulates the gluconeogenesis in all forms. So the same thing has been shown here, that when we take food, the food will create the hyperglycemia. That hyperglycemia is sensed by beta cells because all beta cells, the glucose will go into beta cell when the blood glucose level is high and the glucose inside the beta cell is less, and that is done by basically the transporters GLUT1 and GLUT2, and the beta cells will utilize that glucose and make the ATPs, and these ATPs will work on the potassium, the ATP sensitive potassium channels, potassium and these channels when they are closed. Then what will happen that the depolarization will occur, and that depolarization will definitely create the change in the calcium channel, and they open because they are voltage gated, and the insulin release will occur. That insulin in liver converts glucose to glycogen, and the glucose level will go down, and not only that, the insulin will act on other cells as well, so the glucose level will go down. Whenever the glucose level goes down in the fasting condition or in other conditions, then the alpha cell will secrete the glucagon, and glucagon will again take care of things and convert glucose, the glycogen into glucose, and the glucose homeostasis will be maintained. MRNA is another hormone which is secreted along with insulin, together with insulin, and co-secreted in response to glucose as well. In supraphysiological levels, it promotes the development of insulin resistance. Not only that, the mRNA participates in glucose homeostasis by two mechanisms: retarding gastric emptying dose response manner, that in supraphysiological doses the mRNA will decrease the gastric emptying, and it also suppresses the glucagon secretion. The neural regulation of blood glucose, which is more complex, is basically done by the counter-regulatory hormones released in stressful conditions: incretins, leptin, adiponectin, and neuropeptide Y. When we talk about all of them, then you see that whenever the hormonal and the various nutritional signals, they are continuously reaching to the brain, and in the brain basically everything is controlled at the hypothalamic level, and in hypothalamus various centers are responsible for it, and hypothalamus controls the liver, and the liver, which is responsible for the hepatic output of glucose and also the storage of glucose. This all is controlled by hypothalamus by various neurotransmitters and also by the autonomic nervous system. Also to the beta cells, again by the neurotransmitters and autonomic nervous system, and hypothalamus also has the control over skeletal muscle, and that by that control skeletal muscle can utilize more glucose or less glucose. Also the white adipose tissue. So all these liver, beta cells, skeletal muscles and white adipose tissue, they take part in glucose homeostasis as well as lipid homeostasis under the control of hypothalamus. While the brown adipose tissue is basically responsible for energy expenditure, or you can say it is controlling the energy homeostasis rather than the glucose and lipid homeostasis. All these centers at the hypothalamus, they are further important for the energy intake or food intake, and various centers are responsible for hunger and other important vegetative reactions. So neural regulation is part of the ominous octet. That is why we are concerned about it. That in ominous octet we know that 8 pathophysiologies, different 8 pathophysiologies. One of that pathophysiology is the neural regulation, and we also know that any dysregulation in that particular neural mechanism is also responsible for the development of diabetes. That is the one of the important part of the ominous octet. But this neural regulatory mechanism yet not have been targeted for the treatment. We do not have any novel agent which can address this, particularly this particular pathophysiology. We know that we have almost drugs for all other 7 pathophysiologies, including the SGLT2, latest one, to address the kidney regulation. So regulation via incretin can also be set a neural, because we know that the glucose dependent manner, the incretins are controlling the blood glucose, and we know that whenever glucose reaches into the intestine, then the GLP-1 and GIP are secreted by the L and K cells of the intestine, and both these hormones on glucose dependent manner act directly on the beta cells and can alter the secretion of insulin, and they do have other central effects via GLP-1, and these central effects will affect the appetite. They will also alter the control of the gastric emptying, and by these mechanisms glucose homeostasis will definitely be altered. So one of the neural mechanisms via incretin, and we have the drugs to cover that pathophysiology or change in the pathophysiology which has occurred because of the advent of the type 2 diabetes. So that particular neural regulatory mechanism is being addressed by the various drugs. So more pronounced in pathological conditions, these all neural mechanisms have been seen that in physiological conditions they have good homeostasis mechanism, but in pathological conditions these factors become more pronounced in comparison to physiological conditions. So first I will talk about epinephrine. Epinephrine secreted from adrenal medulla and released in response to exercise and decreased blood glucose, both conditions. So every stressful stimuli and also the decrease in glucose concentration will release epinephrine, and it will stimulate liver and muscle phosphorylase, and that will produce the glycogenolysis as well as new glucose formation by neoglucogenesis. Increases liver glucose output by both mechanisms and muscle glucose metabolism as well, because muscle do have glycogen. So that epinephrine will be secreted, and this for the safe response, and in the fasting condition when the blood sugar level goes very low, then this response should occur. But a constant stressful condition by this particular mechanism can also produce adrenal diabetes, which we refer as adrenal diabetes because it is because of the cortisol mechanism. And we can see here that how epinephrine is working on the G protein coupled receptor via the G protein, the adrenal cyclase, and the second messenger CAMP, the protein kinase A, and finally inhibiting the glycogen synthesis as well as promotion of glycogen breakdown. By both these mechanisms, it will maintain the homeostasis of the glucose. And here it has been shown that how the various other hormones, not only insulin, the leptin do have the effect, and acting via the various mechanisms which are involving other neurotransmitters as well from the brain, and that controls other glucose homeostasis ways by decreasing and increasing the changes in the insulin level, insulin sensitivity, insulin resistance. And this leptin is reaching to the brain via the fat cells, via the involving in that fat cells. And not only that, by this mechanism, alteration in the various metabolic activities in the liver and the fat tissue do occur. And here it has been shown that how the adipose tissue, by acting on the blood vessels, via the blood vessels, the glucose, insulin and free fatty acids can have effect on the gut, and sensing at that particular level at gut can reach to the brain, and brain can have the action via liver, via pancreas, via skeletal muscle. By that, in liver it can decrease glucose production, increase glucose uptake, the formation of glycogen, and increase glycogen storage. And in pancreas, by acting on insulin and probably by amylin as well, and in skeletal muscle, influencing the glucose uptake, the homeostasis can occur. The neural regulation can also be seen here again, the same thing, that how meal can have the impact on gut, and that gut can release the GLP-1, which is mainly released in the presence of glucose, and how the GLP-1 can have effect at the various levels, at the level of liver and at the even sensing and augmenting at the level of brain as well, and how that can augment and work on these three particular liver, pancreas and skeletal muscle, and by that glucose can be regulated. Leptin is another hormone which is secreted from adipose tissue. Although low levels of expression have been detected in but they are also seen in the hypothalamus, hypophysis, placenta, skeletal muscle, stomach epithelium, and even the breast. The leptin circulates bound to a carrier protein, and its level increases as a function of the fat mass. It interacts with specific receptors located in the particular nucleus, which we can say at the level of medulla, and demonstrated by the anaphylactic effect achieved by its local injection and its lack of effectiveness when the air is disrupted. So by that we can say the leptin inhibits insulin secretion and increases insulin hepatic extraction. So it's acting at both ways. Activates sympathetic tonus, by that again the autonomic nervous system, by that again it can augment the release of insulin. It also decreases hepatic glucose production, increases insulin sensitivity and decreases glucagon levels. So by acting on the glucagon levels as well as on insulin levels, it will have impact on the glucose homeostasis. Leptin circulates bound to a casein protein and its level increases as a function of the fat mass. It interacts with specific receptors located in the AN and demonstrated by the anorexic effect which we have discussed. Adiponectin is exclusively expressed in and secreted by white adipose tissue. High circulating level of the protein are present in human plasma as a polymer of 18 monomers, and adiponectin acts as insulin sensitizer. Its plasma concentration decreases in obesity and in type 2 diabetes. Administration of recombinant adiponectin to rodents increase glucose uptake and fat oxidation in muscles, reduce fat acid uptake and glucose production in liver and decreases insulin resistance. So its impact on insulin resistance has been proven by various experiments. In rhesus monkey, decreased circulating level of adiponectin are associated with the development of insulin resistance and type 2 diabetes. So it has been associated with IR. In mice, the drug like the thiazolidines not only increase insulin sensitivity but also plasma levels and mRNA production of adiponectin, that we know. Because of that particular fact we are using these particular drugs. Also the telmisartan has been shown to have the impact on the adiponectin level. At clinical level there is a negative correlation between adiponectin and body weight and body fat mass and insulin levels. The neuropeptide Y, this peptide is produced by neurons located in the floor of the third ventricle, and neuropeptide Y injection has already shown this all effect in the animal studies, and because of these actions we can say that NPY administration will produce obesity. So it has got some role in energy homeostasis as well as in glucose homeostasis. May be acting via the insulin only. So this very complex slide again shows the same thing, that the three organs basically liver and the fat cells and the pancreas and muscle are involved in the various mechanisms which are initiated at the level of the brain and by various new neurotransmitters. These all regulations are occurring are controlled at the level of brain, and basically they do alter the glycogen synthesis or glycogenolysis or insulin sensitivity or insulin resistance. Insulin and brain. The brain is an insulin sensitive organ, we all know it, and various studies have shown that insulin action in the brain affects energy and glucose homeostasis, neuroendocrine function, reward, memory and learning. Neurons in many areas of the brain express insulin receptors, and insulin acting on these receptors causes activation of the various enzymatic signaling pathways, and by that insulin mediated activation of these pathways can lead to short-term changes in neuronal activity or prolonged changes in gene transcription and neuronal plasticity. These are chronic changes really occurring in a diabetic person who is deprived of the insulin. More important in pathological conditions rather than physiological conditions. This particular action of insulin in the brain, the CNS insulin to relation to the production of glucose in the liver regulation might however be somewhat greater in the setting of insulin deficient type 1 diabetes where insulin is delivered systemically. Because in type 2 diabetes whenever we replace insulin, it is basically in type 1 and type 2 both systemic and not the physiological. Physiologically we know that insulin is reaching liver first and then goes to other tissues, other cells. But when we are replacing insulin, it can never be physiological because liver is getting the last bite. Brain insulin signaling does appear to exert important tonic, direct and indirect effect that serve to limit successive glucose production. Thank you for the patient.